This is the second video for the cellular respiration unit. In this video, we will be discussing the first three pathways of cellular respiration, which include glycolysis, pyruvate oxidation, and the citric acid cycle. There are 4 different stages of cellular respiration. It will be important for you to understand what the main substrates and products are from each stage, including the amount of ATP and electron carriers produced. The first stage of cellular respiration is called glycolysis. It occurs in the cytosol of the cell and is considered anaerobic, meaning it does not require oxygen. Glycolysis involves the oxidation of glucose, a 6-carbon sugar, to pyruvate, a 3-carbon sugar. Pyruvate is then transferred into the mitochondria, where stages 2 through 4 will occur. These stages are all considered aerobic. They require oxygen in order to occur. The second stage of cellular respiration is called pyruvate oxidation and is a single step where pyruvate is oxidized to acetyl-CoA, a 2-carbon sugar. Acetyl-CoA is then completely oxidized to carbon dioxide through the third stage, the citric acid cycle, also called the TCA cycle. The fourth stage of cellular respiration involves an electron transport chain embedded in the inner membrane of the mitochondria. We will talk in detail about this stage in another video. Because stages 2 through 4 occur inside the mitochondria, it is important to understand the mitochondrial structure. It contains both an outer membrane, seen here in dark orange, and an inner membrane, seen here in light orange. The inner membrane contains many folds called cristae, which increase the surface area of the membrane. Between the two membranes is the intermembrane space, and the space inside the inner membrane, where steps 2 and 3 occur, is called the matrix. Glycolysis, the first stage of cellular respiration, is a series of 10 chemical reactions, starting with glucose and ending with 2 molecules of pyruvate. Glycolysis can be divided into 3 phases. The first phase is the preparatory phase. In this phase, 2 molecules of ATP are required to phosphorylate the glucose molecule. The addition of phosphate groups helps trap glucose inside the cell and makes the molecule unstable, allowing the second phase to occur. The second phase is the cleavage phase, where the 6-carbon ring structure is broken into 3 carbon molecules, as seen highlighted in blue and green here. The third phase is the payoff phase, which produces 4 molecules of ATP, 1, 2, 3, 4, and 2 molecules of NADH. This means that glycolysis has a net yield of 2 ATP molecules because 2 of the produced ATP are canceled out by the 2 ATP that were spent in phase 1. The second stage of glycolysis involves the conversion of the 3-carbon pyruvate to 2-carbon acetyl-CoA. This produces 1 molecule of NADH and 1 molecule of carbon dioxide. The carbon dioxide is produced from the carbon that is removed from pyruvate to go from 3 carbons to 2 carbons. Once produced, acetyl-CoA will then enter the citric acid cycle. Sometimes you will hear the citric acid cycle referred to as the Krebs cycle or the TCA cycle. During this stage of cellular respiration, the fuel molecules will be completely oxidized. First, the 2-carbon acetyl-CoA is joined to a 4-carbon oxaloacetate to form 6-carbon citrate. A number of steps then occur to regenerate the 4-carbon oxaloacetate, which is why this is called a cycle. In the process, 2 molecules of carbon dioxide are released, along with 3 molecules of NADH and 1 molecule of FADH2. There is also 1 molecule of ATP produced through the process. The citric acid cycle also plays an important role in anabolic processes, as some of its reactants can be funneled off into other pathways to build various macromolecules in the cell. These anabolic pathways require energy. In summary, the first stage of cellular respiration, glycolysis, takes a molecule of glucose and converts it to 2 molecules of pyruvate. In this process, we produce a net yield of 2 ATP and 2 molecules of NADH. The 2-pyruvate will then flow into the next stage, pyruvate oxidation, and produce 2 molecules of acetyl-CoA. This process will produce 2 molecules of NADH. Remember, all the values are doubled because we had 2 molecules of pyruvate enter, so we are producing 2 molecules of acetyl-CoA. Those 2 acetyl-CoA would then flow into the citric acid cycle, where they will be oxidized to 4 molecules of carbon dioxide, and in that process we will produce 6 molecules of NADH, 2 molecules of FADH2, and 2 ATP. Again, remember all the values are doubled because the citric acid cycle will be running twice, one for each of the acetyl-CoA. At this point, we have produced a total of a net yield of 4 ATP, 10 NADH, and 2 FADH2. The ATP produced has been through substrate level phosphorylation. While the remaining energy is stored in the electron carriers. These molecules will now go to the electron transport chain, where their stored energy will be converted to ATP through oxidative phosphorylation.